Hello again. This is Dr. Beall again, and this lecture is going to be about the biochemical aspects of aging. There is a chapter in the textbook on aging, but aging is a highly studied and rapidly changing field that doesn't lend itself well to being captured in a textbook. I have based this lecture more on the framework of this article that I have listed here, The Hallmarks of Aging, that was written by Lopez, Oteen, and others. And I think they do a good job of systemizing this complex and diverse area of research. And that article has been then adopted by many other authors as a framework going forward. So here are the topics that I will touch on. First, I will talk about some general aspects of aging science, including the relevance to dental practice and the concept of health span as opposed to lifespan. I will then introduce the hallmarks of aging framework with the 9 hallmarks that those authors identified. I will discuss the loss of proteostasis and how that possibly relates to Alzheimer's disease. Then I will talk about nutrient sensing and the phenomenon that caloric restriction can reduce aging in experimental animals. Then I will cover some additional hallmarks, including genetic instability, cellular senescence, and telomere attrition, along with a couple of the other ones. And finally, I will discuss oxidative damage, which is not one of the identified hallmarks, but relates to several others. How does the phenomenon of aging relate to dentistry, and what do dentists need to be concerned about? And there are a number of things to discuss in that way. One has to do with medications. Many older patients will be on medications, often multiple medications, which is sometimes referred to as polypharmacy. Those can affect dental practice. For instance, a common side effect of many medications is xerostomia, which can predispose to dental caries. Another medication effect is that bisphosphonates can be related to osteonecrosis of the jaw following dental procedures. Chronic periodontitis increases in prevalence with age to a great degree. It's not entirely clear if it's mechanistically related to aging or if it is just a disease that takes a long time to develop and then appears in older people, but it does involve inflammation, and inflammation increases with aging generally, so there's probably a relationship there. Also, oral cancer is another condition that like most cancer, appears more in aging patients. So that's something that the dentist would have to be concerned with. Another aspect of dentistry that is affected by aging is the risk of systematic infections will go up with age. Dental patients can experience a transient bacteremia following dental procedures, and the released bacteria can cause opportunistic infections elsewhere in the body. And body sites that are especially susceptible are the heart valves, especially when they have certain defects, or artificial joints. And situations like those are more common in older individuals. So that's something that dentists will have to look out for. It may be best practice to give prophylactic antibiotics to prevent such infections. Finally, dental problems can detract from overall health, which brings us to the concept of health span, which is the subject in the next slide. So, the dental health is so increasing the health span means, keeping dental health as long as possible too. And the idea of health span is that there is a period of life where you're not suffering from age-related disease, and then a variable period at the end of life where the quality of life is low because of aging, because of the age-related disease. And the health span is then that part of the lifespan that is free of severe disease. And it's really what one would want to extend, as opposed to just extending lifespan and having a long period of ill health at the end of life. So that's sort of shown on the left-hand part of this slide, that optimally you want to just have the health span be as much a part of the lifespan as possible. And on the right-hand part of the slide is just a survey from the Pew Research Center that actually suggests that the problems of old age might not be as bad as younger people think, that younger people are expecting to have all these problems and older people are really not experiencing them. To go back to the lifespan, an early mathematician and actuary named Benjamin Gompertz noticed that you could describe survival rates of different populations by their death rate. And that death rate increased exponentially by age, and that's called the Gompertz equation. So on the left-hand plot is shown that for the species of mice, dogs, and humans, and so they each have a line in terms of their death rate is going up by age, but it goes up faster for mice, intermediate for dogs, and then less for humans. The right-hand plot shows that for Drosophila, the equation varies according to which temperature they are raised at. So the death rate goes up slowest at 20 degrees centigrade, and those flies live longer, and then it goes up intermediate at 25 degrees and highest at 30 degrees. So in any case, the death rate is exponentially related to age, and it's expressed by that equation that's shown on the left. However, it turns out there's a possible exception to Gompertz's equation, which is the naked mole rat. This is a rodent that lives in underground colonies in East Asia and East Africa, and it's actually a eusocial, so it's more like has structured. Those colonies are structured more like bees and ants than any other. It's the only mammal that's known to have this kind of social structure. And Columbus Zoo used to have a colony of them, although I don't believe they do anymore. Anyway, it was known that they could live a long time, up to 30 years, which is the longest lived rodent. Researchers looked at their age-related mortality, and you can see in the green line in those graphs on the right that it doesn't seem to increase over age, and that's different from the humans, mice, or horses, which are also shown in those graphs. And in the graph in panel E, then they've sort of tried to put them all on one scale by setting one as the age at which the different organisms are sexually mature. Although where they were setting it for horses didn't seem quite right, but that's a different story, I guess. But so you can see that the naked mole rats are now an interesting system, and they're being used in aging research to try to find out why they're different from everything else. So just in considering the general state of aging research, then it's a field that's very hot. There's a lot of work in it with people have and controversies, and people have been fascinated by the idea of immortality or of regaining lost youth, forever and ever, and both in the wider culture, and scientists are feel the same things, and that feeds people to want to work on it, and also brings it a lot of media attention and hype and controversy, and it's been fast moving, and it makes it hard for textbooks and so forth to keep up with, so as I mentioned, I'm going to largely follow this framework of hallmarks of aging. And the authors of that article give 3 criteria for what they're defining as hallmarks, and they're trying to sort of systemize this very complex field. So their criteria are they must be things that manifest during normal aging. The experimental aggravation of whatever the hallmark is should accelerate aging, and amelioration should retard the normal aging process and increase the healthy lifespan. So those are the three criteria. And then they've identified 9 hallmarks here that are shown in this circular graph. And they aren't, standalone kind of things. They do interact with each other, and we'll see some of the ways that the things interact. So the 9 hallmarks are genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. A salient fact is that these are not isolated factors. We will see in some cases how they interact. And for instance, that telomere attrition might cause a cell to become senescent, and that might in turn cause the cell to secrete cytokines, which causes altered cellular communication. And we'll see that. And there are many other examples along this line. So to sort of move on to those hallmarks then, we'll start with the loss of proteostasis. And this is generally due to adverse effects on proteins in the cells. And here's this diagram here. Here you can see that there are these sources of damage that can affect a folded protein. And it can be heat shock or stress in the endoplasmic reticulum or oxidative stress, and it can affect the folded protein, and it can cause it to unfold. And that's bad because, as you hopefully know, proteins need to be folded into their specific three-dimensional structures to carry out their functions in most cases. And a number of things can happen to the unfolded protein then. Some of them are good and some are bad. One is that it can be targeted to the lysosome by a couple of different mechanisms, and is then degraded by what is called autophagy. Another form of degradation involves first covalently attaching multiple copies of a protein called ubiquitin, which targets the misfolded protein to a structure called the proteasome, which I believe was mentioned by Dr. Bartling in his lectures. A third process that can occur involves heat shock proteins, or HSPs in this diagram, which participate in chaperone-mediated refolding of the protein to its correct native structure. So those first 3 outcomes are basically favorable, but the fourth outcome is possible, in which the protein aggregates, which some unfolded proteins have a tendency to aggregate. And that's the outcome that can contribute to aging. So a disease of aging that's associated with loss of proteostasis is Alzheimer's disease. As I'm sure we all know, it's a progressive disease with memory loss being a primary symptom. That picture is of a normal brain on the left and an Alzheimer's brain on the right, and you can see macroscopic effects in the Alzheimer's brain, such as enlargement of the ventricles and shrinkage of the hippocampus, which is important in memory formation, and shrinkage of the cerebral cortex that has a diverse array of sensory, motor, and associative functions. A couple of microscopic abnormalities have also been found in the brains of Alzheimer's patients. And the first is amyloid plaques. They're called amyloid because they stain with a compound that also stains amylose or starch. But they aren't starch, they're actually protein, as we'll see. And researchers were able to identify what they are composed of, and it's called a beta amyloid peptide. And we will get into more detail of that. The beta amyloid plaques are extracellular. The other microscopic abnormality is called neurofibrillary tangles, and they are made of a microtubule-associated protein called tau protein, and they are located intracellularly. These are micrographs of the amyloid plaques on the left and neurofibrillary tangles on the right. And you can see an artistic version of the two. And so you can kind of correlate, where within the cells they are and what their appearance is like in a section. This shows an artist's view comparing Alzheimer's to normal neurons within a single view. On the right is the normal cell with the transmembrane protein, amyloid precursor protein. In the healthy case, it is cut by two proteases. The gamma secretase complex that includes the protein presenilin and the alpha secretase in yellow and green. That releases a protein called soluble amyloid precursor protein alpha, or SAPP alpha, into the extracellular space. The healthy neuron also contains healthy microtubules and healthy mitochondria. On the left is a situation in Alzheimer's disease. In that case, the APP is cleaved with gamma secretase, as in the healthy case, but also the beta secretase, as shown here. And that cuts in a different location and releases the soluble APP beta and beta amyloid, which we've heard is part of the plaques. The beta amyloid then forms into oligomers, then fibrils, and then finally the amyloid plaques. You can also see in the disease neurons that there are disintegrating microtubules that are releasing tau fragments that aggregate into the neurofibrillary tangles, and also there are damaged mitochondria. The details of how the mitochondria relate to other things are not totally clear yet, but it is known that that Alzheimer's brains use glucose less efficiently than healthy brains, and there are other studies showing various defects in the mitochondria in Alzheimer's as well. So they are shown here. Now, there are actually two forms of Alzheimer's that can be distinguished. So the first form is fairly rare and has a strong genetic basis. It's called familial Alzheimer's disease. It occurs in younger people generally than the more common form. And there are two major types of mutations that have been found that cause the familial form. And they are genes that are related to the production of amyloid beta. They are in the amyloid precursor protein and in genes called the presenilin genes 1 and 2 that are part of the gamma secretase that we just saw. So, they're basically involved in that pathway. The other form, sporadic Alzheimer's disease, is the more common form and occurs later over 65 years of age. So the causes of that are uncertain, but there are a few genes that increase the risk. The most prominent one that increases the risk of sporadic Alzheimer's disease is the apolipoprotein E, and the ApoE4 allele in particular, that increases the risk by around fourfold, or maybe even more if you have two copies of that allele. And other genes have been found that have somewhat less risk for the sporadic form of Alzheimer's disease, but nothing like the familial form where it's clearly these mutations and in specific genes that are involved, seem to be involved in this pathway. So this is showing kind of the same thing that was shown in the previous slide about APP and the cleavage, but just showing it a bit more clearly than that last slide. So the intact APP is in the middle of this slide, and going to the left is the pathway with alpha secretase that does not lead to beta amyloid, and while the pathway to the right has beta secretase and does lead to beta amyloid. And then I mentioned again that the mutations in the familial Alzheimer's disease are in the APP itself and in the gamma secretase subunits of the gamma secretase. So from those mutations and the fact that beta amyloid peptide is in the plaques that are characteristic of Alzheimer's brains, there's a strong hypothesis that beta amyloid production and aggregation are the driving force for Alzheimer's disease. And the role of amyloid has also been supported by mouse models, like making mice that have the familial Alzheimer's disease mutations in those proteins. But there are also quite a few results that do not agree with this simplest form of the beta amyloid hypothesis. So one contrary evidence is that humans can have the plaques without disease, and it was made possible to discover this by a method using positron emission tomography, or PET scanning, that lets researchers visualize plaques in living people. Previously, that had only been possible in brains from autopsies. So they could see plaques in some people that did not have the disease. And this method also was able to find that in some patients with the disease, sometimes you can reverse the plaques without curing the disease. It was also, although they had these mouse models that were had mutations in prenicillin or APP, that they found those mice did not get the tau tangles that you find in the human disease or the irreversible neurodegeneration. It was always reversible, whereas it doesn't seem to be in humans. And in terms of the brain locations that are affected by the disease, they tend to be more the regions where the tangles predominate as opposed to the amyloid plaques. And that's something that doesn't fit well with the straightforward amyloid hypothesis of Alzheimer's. And finally, and possibly most important, drugs that target amyloid plaques have not been successful in trials in humans. I saw one estimate that around 400 drugs have been tried and only a few approved, and none of those still are especially effective in treating Alzheimer's disease. So one outcome of this whole story is that there's been more work to get improved animal models to effectively test drugs. And a broad picture of things is that there's clearly evidence that amyloid is important, but some gaps in our knowledge are still around, including how amyloid affects tau and leads to tangles, and whether it can cause irreversible changes that persist even when the plaques are removed, and what the role of mitochondria are. And there are still many questions that are left, and we still don't really have good treatments for Alzheimer's. So now I'm going to talk about one of the other hallmarks was deregulated nutrient sensing, and how that led to aging. And that sort of dates back to a finding that, an early finding in aging research that is still sort of bearing fruit, that caloric restriction could increase the lifespan of animals. And this actually dates back to 1935 and was first shown in rats. And it has also been found in mice and flies and worms, and other organisms as well. This shows an experiment from 1986 that was reproduced in the textbook. That is, and so to sort of give it a little background, generally when you have mice in the lab, you just give them more food than they can possibly eat, and just leave it in sort of sits on top of the cage, and they just eat whenever they want, which you call ad libitum feeding. And then you can use that and measure what average mice will eat under those conditions. And then you could give mice less than the amount that they would eat if they could eat as much as they wanted, and reduce that by a certain percentage. And that's what they do to these different populations of mice. And then here in this experiment, they've either not given them as much as they wanted, or reduced it by 25, 55, and 65%. And then this is a survival curve, which is just the percentage of the starting group of mice that is alive at various months of age, as you see here. And you can see that the restricted mice are living a lot longer. So, the longest lived, they're on average living more than a year longer than the ones that are eating as much as they want. And that's for an animal that only lives, a little over 2 years normally. So that's, pretty huge increase of lifespan for the mice with this criteria. Now, so people have wondered, it works in these flies and worms, but would it work in humans? And someone did try it on monkeys, which gave partially positive, but it wasn't really strong. In fact, it turns out to be very hard to do on anything like an animal like that. So there's a couple graphs shown in here that from this experiment on monkeys, on macaque monkeys. On the left side is a timeline of the calorie restricted and controls groups, with deaths marked as the gray hatch marks, and then on the right there are the survival curves. So one thing you can see from the timeline sort of thing is that they carried out the experiment for 20 years. So that's really a tough experiment to do, and monkeys are not cheap to take care of, basically. So it's a hugely expensive experiment too, and you can see they had to add more groups after 5 years, because I think they realized the numbers were not going to be high enough to tell if it was extending life or not. And then the reason why there are two groups of survival curves shown is because they had they excluded some animals that they claimed died for non-age related reasons, which is not ideal. And so in the overall mortality curve, you can see that it doesn't show much difference. But if you exclude these ones that they died for other reasons potentially, then you get more of a difference. And so basically the indication is that science can be really messy. A further point is that when caloric restriction works, though, in these mice and so forth, it lessens age-related diseases such as cancer, as well as extending the lifespan. So it's hopefully extending the health span as well, which we mentioned is a goal of any intervention you would want to make. Researchers have worked out some of the signaling pathways that are leading to the life-extending effect of caloric restriction. And in this figure, they're calling it dietary restriction. And I get the chance to talk about more kinases. That's fun. At the risk of stating the obvious in diagrams like this, the arrows are a positive effect and the bars are negative effect on the next thing in the pathway. So one pathway that dietary restriction inhibits involves the molecule insulin-like growth factor 1, IGF-1. As an aside, IGF-1 acts through a tyrosine kinase receptor, which I told you about in the signal transduction lecture. IGF-1 also acts through PI3K, which phosphorylates phosphatidylinositides at the 3 position of inositol, whereas the IP3 that I talked about previously is the 1, 4, and 5 position. So this is a little bit different. And it signals in a different way than we talked about earlier in signal transduction. The IGF-1 pathway integrates with a couple of other pathways, one of which goes through the AMP-activated protein kinase, or AMPK, and they all come together at a protein called mTOR, which stands for mammalian target of rapamycin. And rapamycin, by the way, is an immunosuppressant that is used to prevent transplant rejection. So mTOR was discovered as being what that drug was acting on. And you're probably not going to believe it, but mTOR is also a kinase. So you've got multiple kinases again in this. Finally, another pathway goes through a protein called SIRT1 for sirtuin 1, and I'll get into that more next. And then there are a few ways that are possible to manipulate these pathways, possibly to reduce aging effects. So high AMP can stimulate the AMPK. High NAD plus can stimulate SIRT1. Metformin, which is a commonly used drug for type 2 diabetes, stimulates AMPK as well. And rapamycin, as mentioned, inhibits mTOR. And so all those could possibly, be things that would fight against aging effects of that are related to caloric restriction. So now we're going to talk about this sirtuins. What are they and what are they doing? And so firstly, they're a family of genes and proteins that are related to a gene called silent mating type information regulator 2, or SIR2. That's a yeast gene. And then from SIR2 you get the family called sirtuins. See, they've kind of like put the 2 in there as well, and that's how the name comes about. But they're a family of proteins, and the members of the family that we're considering here are NAD plus dependent deacetylase proteins. Remember from gene expression lecture how histone acetylation is important in regulating gene expression and causes those epigenetic effects. So that's where SIRT1 is getting involved in as well. And it's removing the acetylation. There were initial reports that sirtuin overexpression can increase longevity in multiple species, and it was also found that the compound resveratrol could bind to SIRT1 and activate it. Also, since it's stimulated by NAD+, the theory is that it might be boosted by nicotinamide precursors. There have also been some contradictory reports, but there are now companies making attempts to boost this pathway. through these supplements, which are these nicotinamide riboside and nicotinamide mononucleoside that may boost the NAD plus levels in cells and possibly fight aging. And those have not undergone clinical trials, but they're available as a dietary supplement where it's kind of an unregulated market. So another of the hallmarks of aging is genetic instability. Mutations in organisms, if they have any effect, are extremely likely to be deleterious. Most mutations are bad, and mechanisms have evolved to make DNA replication as accurate as possible and to repair the DNA if it gets damaged. However, there are trade-offs, and it has been found that the longer lived the organism, the more DNA repair activity it possesses, and that's shown in this graph that's from the textbook. Another piece of evidence that genetic instability is very important in aging comes from what are called progeroid syndromes. Those are genetic diseases that have some signs of premature aging, like cardiovascular disease appearing very early in that are normally only appear in older people, or hair loss occurring at a very early age. And those are often in genes related to DNA repair or maintenance. And examples are the Werner and Bloom syndromes are caused by mutations in DNA helicases, which are enzymes that unwind the DNA strands and allow repairs to be made to damaged DNA. Hutchinson-Gilford syndrome is caused by mutations in a molecule called prelamin A, which is a gene that acts to organize the structure of the nucleus. It goes on to form nuclear lamin, which keeps the shape of the nucleus. And Cockayne syndrome is another progeroid syndrome that's caused by mutations in genes involved in nucleotide excision repair. Another hallmark of aging is mitochondrial dysfunction. I mentioned that mitochondria appear to be affected in Alzheimer's, although all the connections there are not totally known. I won't go through this whole diagram, but a couple of quick points here. One is that DNA damage it can be an initiator of mitochondrial dysfunction, and that mitochondria have their own DNA, and their proteins are coded both there and in the nucleus. Also, reactive oxygen species can cause damage. I'll talk about them a bit later, but they're shown here. And there can be something called a hormetic response, in which a weak toxic treatment stimulates a beneficial compensation, and that can act in this to fight against aging in mitochondrial dysfunction. Okay, this slide shows two more of the hallmarks, stem cell exhaustion and altered intracellular communication. So up top here, the major way that organs renew themselves is with stem cells. And that's a very big area now. I'm sure you've heard about it, where people want to harness them to treat many, many diseases. And during aging, these cells can become exhausted. And here's a list of cells and possible consequences that are aging-related problems. And I've also put there, what the tissues are for those acronyms of the different stem cells. And below here are ways that altered intracellular communication can contribute to aging. One is that aging predisposes to inflammation, and this is even generated, this portmanteau term here shown, inflammaging, and that contributes to many of the major aging-related problems that I listed there. But I want to point out particularly, periodontitis, as I mentioned that in the beginning of the lecture too, that's involved with inflammation. Inflammation is occurring, and so during aging, that would be something that might come up. Okay, the next hallmark I'll spend some time on is cellular senescence. And that was discovered, it's considered to be discovered by Dr. Hayflick, who's shown in the picture here. He discovered something called the Hayflick limit. And that is the phenomenon that you can take cells from an animal, so-called primary cells, and you can put them into tissue culture dishes, especially fibroblasts that make the connective tissue. They'll grow out from, if you dice up some tissue and put it in a dish, and they'll divide about 60 times, but then stop. And that's called the Hayflick limit. And cancer cells, or so-called transformed cells, which are kind of the culture version of cancer cells, don't respect that limit. They just keep on growing, pretty much indefinitely. But for primary cells from an organism, this is true. They'll divide about 60 times and then stop. And Hayflick did various experiments, showing that they were in fact counting the number of total divisions, and somehow in this process. And here's some things that we know about cellular senescence. It can be initiated by cellular damage. It helps to prevent tumors. If the normal program is to stop dividing after a certain number of divisions, tumors have to get around that block. So cellular senescence is a natural kind of anti-tumor effect. And so another beneficial effect of cellular senescence is in wound healing. I'll show that a little bit later. They make an important growth factor. And it has some functions in embryonic development as well. Sometimes cells become senescent, in a programmed way during embryonic development. Lastly, there are some genes that are expressed in senescent cells that can be used as markers to say a cell is senescent, although they vary a little bit depending on the cell type. So this seems like a pretty benign thing. Why is it a hallmark of aging? Because we're saying it contributes to aging, right? That's part of being a hallmark. One thing is that these senescent cells accumulate in aged and damaged organs, and they're found more in certain aging-related diseases, including osteoarthritis, atherosclerosis, osteoporosis, and type 2 diabetes. Senescent cells adopt something called SASP, for senescence-associated secretion phenotype. They secrete various cytokines, growth factors, and proteases, including things that can stimulate inflammation or aid in tumor growth. So this is kind of feeding into that inflammation and cellular communication part of Hallmark. And finally, I'll just mention that there are fairly new drugs that are being called senolytics, that can kill off senescent cells that have shown effectiveness in mice and are beginning to be tested in humans. And it's possible in some cases those kind of drugs could treat multiple aging-related diseases at once, which would be a very nice property if they, and it would avoid some of this polypharmacy that aging people experience. Okay, this picture is a little late for Halloween, but there's a diagram that compares these senescent cells to zombies, and some kind of there are 4 panels there, and some kind of damage induces senescence in the first panel. Then in the second panel, you have the cells, that SASP that I talked about, the secretion phenotype, and then in some cases the immune cells can remove them. That's in the third panel. That's sort of the normal case that you're hoping for. But in the last panel, if you have too many of them accumulate, maybe the senolytic drugs can or they'll be the zombie killers to kill the zombie cells, and that's that little figure. And here's just another diagram that fleshes things out a bit more. Here are stimuli that can lead to senescence. There are a few different things there. Often the cells that become senescent, the other choice would be to undergo apoptosis. If they get damaged, that's the term for programmed cell death. You've probably heard that. And what the senolytic drugs are doing is pushing cells out of the senescence pathway into the apoptosis pathway. So that's how they're killing the cells, is just by changing that pathway to say, oh, you want to go kill yourself, actually. And these are some of the markers that I mentioned that are elevated in senescent cells that people are using to, try to count the number of senescent cells in various, diseases and so forth, and show that they're elevated. Then there's some beneficial aspects that I mentioned. They're kind of on this side of the pathway, the wound healing and embryonic development and so forth. And that they can be cleared out by the immune system, and everything's fine then, as well as another pathway besides the apoptosis. And then this just shows that there can be deleterious effects if too many senescent cells build up in a tissue, and the kind of deleterious effects are shown in this pathway and lead to aging, and they also get resistant to apoptosis on that pathway as well. So the reason why Hayflick's cells were becoming senescent was probably because of this other hallmark of aging, that's telomere attrition. And Dr. Lies, in his lecture on DNA replication, gave a bit of this background, so I'll sort of speed through it. But he was talking about, he mentioned that replicating to the end of a linear DNA, like a mammalian chromosome, is a problem on the lagging strand because you don't have room for priming for the last bit of it. You're making all these Okazaki fragments and ligating them together, starting with RNA primers, and the RNA primers have to get removed, and so at least that is going to be lost. Plus, you need some room to prime and everything, so you can't get all the way out to the very end, and so you need telomerase to keep extending the ends if you want to keep the ends from shortening. And here's telomerase. He mentioned it contains an RNA, that's part of the telomerase enzyme, the green thing, and it can template itself and make these short repeats that are the ends of the chromosomes, the telomeres. And so the thing about telomerase, it's active in germ cells and in many tumors, but it's turned off in many somatic cells. And that's probably why Hayflick cells would not keep dividing, because they didn't have the telomerase. The telomeres get short over, many divisions, and eventually they become senescent. But if you can actually, use gene manipulation to put the telomerase gene into these, fibroblasts and whatever primary cells, and then they will in fact grow in culture pretty much forever. So it's the telomerase being missing or not active is really what's keeping those cells from in the Hayflick limit. For the last few slides, I'll discuss a bit about oxidative damage and aging, and this is big in the textbook. So reactive oxygen species, or ROS, are produced either as a minor byproduct of oxidative phosphorylation, or in some cases intentionally. For example, here's by a phagocyte to damage an infecting bacterium. It's producing a lot of hydrogen peroxide and so forth to damage the infecting bacterium. And they are highly reactive, as the name suggests, and can cause damage to proteins or mutations in DNA. And some of the important reactive oxygen species are superoxide radical, hydrogen peroxide, and hydroxyl radical. So it's been found that the amount of oxidized amino acids in proteins increases with age. So that's shown here for oxidized methionine on the left and oxidized tyrosine on the right, that as people get older, they get more and more of this amino acids in their proteins. And Other reactions that can occur with oxidation are production of things called glycoxidation end products and lipoxidation end products. And the general scheme is that free radicals cause amino acids and proteins to react with either lipids or sugars, and that produces these two products. And three of the major ones that are formed have the structures shown here. And here you can see that these products increase with age, those three products. They're comparing the skin in yellow with articular collagen in orange. And in that case, you can kind of see the skin is getting less of those products, which they attribute to the higher turnover rate of skin relative to the joint collagen. At any rate, so there's a whole theory of aging, and this is in the textbook, that the major driving is oxidative damage, and then it's called the free radical theory of aging. And we've seen roles for oxidative damage in some or all of the hallmarks of aging. We saw it in that it could damage the proteins, and that could lead to loss of proteostasis. Damage to DNA can lead to the genomic instability. And, reactive oxygen species also contributed to the mitochondrial dysfunction. But as an overriding theory, there seem to be some shortcomings. For example, there are essential vitamins that are antioxidants, but giving them to animals doesn't extend their lifespan. And also, mice and worm genetics don't seem to show effects of oxidation on lifespan. Some experiments showed that mutations that increased oxidation actually led to increased lifespan, which may be due to the hormetic effect that I mentioned earlier. So, and then down in the corner there, some people are even questioning if that theory is dead. But there's some role to play, but it's, as an overriding theory, it doesn't seem to be quite what's driving everything. So I'll just recap now. That's the final direct slide, and we'll go back over. Aging is a complex field, and there's problems with extending findings from experimental animals. In Alzheimer's, we've seen treatments that work on the mice that don't work in humans, and so forth. There's problems with correlation, versus causation of various things. You see things get elevated in aging, but does it really mean they're doing anything? It may be, and I consider it useful to think of the spectrum of facets as described in the hallmarks, and rather than the overarching theories such as the free radical theory. That's sort of general philosophy kind of thing, and also general philosophy that health span is a useful concept as well as lifespan, and that's really what you would like to extend as much as possible. To move on, Alzheimer's disease seems to involve proteostasis to a great degree. There are amyloid plaques and neurofibrillary tangles. Beta amyloid makes the plaques, and it is processed out of the APP protein, the amyloid precursor protein. And mutations in familial Alzheimer's disease are in the genes for that amyloid precursor protein, and also the secretase gamma, a subunit of the secretase gamma protein. And familial Alzheimer's is something that's strongly, linked to genetics and arrives much earlier than the other form of the sporadic Alzheimer's, which is the other form. And then there's tau is a protein that makes the neurofibrillary tangles, and it's usually a microtubule-associated protein. Okay, caloric restriction or dietary restriction reduces the rate of aging. So, and in that, pathways involving the insulin-like growth factor, or IGF-1, appear to be involved. A pathway with the sirtuin-1, SIRT1, may also be involved. I didn't put it on here, but the mammalian target of rapamycin mTOR is kind of a central player in the pathways for caloric restriction as well. Then there's genetic instability, which the progeroid syndromes cause accelerated aging and affect DNA gyrases for repair of the nuclear lamin and genes involved in excision repair as well. Can cause progeroid syndromes. Okay, inflammaging is a term indicating that aged organisms have a chronic state of inflammation, and that may relate to periodontitis, and it also relates to multiple other aging-related diseases. We talked about cellular senescence. The Hayflick limit describes the fact that somatic cells have a limited ability to replicate in culture. There are senolytics, which are drugs that might eliminate the senescent cells. There's this secretion phenotype that occurs in senescent cells, and they accumulate in damaged and aged organs. And due to telomere attrition is what causes the Hayflick limit because of lack of telomerase. And telomerase is this reverse transcriptase that's really present in germ and in cancer cells and not in all normal cells or expressed in those cells. And the free radical theory is an older theory of aging that is, now coming not fully borne out as an overriding theory. So, and I want to just thank my live audience for this lecture, although she did express the desire to hear more about kinases.